Okay, on that we going to a segment number D on validation. It's going to be just a very brief description of validation now. Validation is a huge topic. People spend tons of time on that, right? So just give you a review of the validation, some pointers from some of the regulatory landscape overview. The reason why we do method validation is because we have to do it. It's required by law. For any of the regulatory testing regulated by the US FDA or EMA, or where we are dealing with life-saving drugs to assure the safety and efficacy of the drug product and drug substance, we have to do method validation to ensure that the method used for critical measurement of some of the critical attributes are validated, right? That actually works. So here are some of the regulatory guidance from the ICH guidelines, which is the Q2R1. And method validation data must be submitted with any regulatory filing, starting from the IND and NDA and any supplement, okay? While method development report may be submitted if you want to, only if they support method validation, right? So method development is not a GMP process. Okay, there's a fine line between verification, qualification, and method validation, right? So in many cases, method qualification and validation are kind of almost synonymous, right? Okay, I also want to mention the regulatory agency endorses any science-based and risk-based approaches to method development, right? And most method development and drug development are phase appropriate, right? Meaning that things become more stringent or more regulatory oversight with later phases, right? And method validation process also is kind of have to be phase appropriate, right? You can always do more, right, in early phases, but why do that if you don't have to, right? And also, regulatory agency endorses the application of QBD principle. QBD stands for quality by design, meaning a science-based, a risk-based approach, and also heavy use of statistics and heavy use of a DOE, design of experiment. Here is a summary of the guidances and resources, right? So all these, they are being documented in chapter 11 of the book, and also the whole process of method validation. Also, I want to show you that today LCGC magazine just published a white paper on analytical method validation, right? It's one of my best white paper that I've ever written. It was actually published today on method validation. Okay, so I'll give you the references later on, right? It was just available for download today. And eventually I will make it available on my website also. Okay, very quickly, most of you are familiar with this process, method validation characteristic, right? So these are some of the characteristics. These are taken from the ICH Q2R1 and also from some of the USP guidance. They're very similar, right? So method validation is very well established principle. Everybody does pretty much the same way, but also everybody have their own detail, right? Have different generic templates. Everybody also do it their own way. And everyone is have different approaches to the detail in method validation, right? But the criteria is very straightforward and well documented. Okay, so here is the parameter explained, right? From specificity, accuracy, precision, sensitivity, linearity. So I won't go into that in too much detail, but just using that as a quick highlight chance to point out some things that you should be perhaps be more aware of. Okay, I want to say that the data element is also documented in the USP 1225, right? Which is dividing the different category of method, right? So stability indicator method actually span the requirement for 3 different types, right? Definitely method category 1 for the API, because we use a composite approach to do both API and also impurity, right? So you definitely have to be in compliant. For late stage validation, you have to verify all these things. We also use the composite method to do identification, because once you do the stability indicating assay, you also identify the API, right, by matching that with the reference standard, right? So you get category 4 by default, right? Without doing any extra work, right? So typically use HPLC retention time plus FTIL for your drug substance and drug products. Okay, so a little bit of method specificity. Typically, we demonstrate it by using a resolution test mixture, which are used for system stability, right? So here's one example where you demonstrate the analyte can be separated from some of the expected impurity declaration product. Here's one example. We show you different way to measure peak purity. I have 2 peaks, right, which looks like well separated, right? The UV spectrum from PDA looks pretty good. They all look kind of very similar, right? But UV does not give you a lot of information, right? However, if you look at the mass spec, right, you can see the first peak. Actually, the mass spec is very different, indicating that there's 2 parent ions in the front of the peak and in the back of the peak, right? So the mass spec actually allow you, just by observation, to go down to 0.5 to 1% level of a co-eluting peak, right? So mass spec is actually pretty good, but it's not good enough for peaks underneath the API peak, right? Because we have to go down to 0.05%, right? So that's why we use a secondary orthogonal method to try to split out any shoulders or co-eluting peak from underneath the API peak, right? So this is to be expected. A case study from Larry Wigman, who's my ex-colleague at Genentech, who gave a workshop, which I'm the organizer of this workshop, right? Showing you how do you do accuracy, repeatability, and range in one study, right? Using a 9 sample preparation, right? 70%, 100%, 130%, right? So these are pretty well expected in the regulation. Okay, so once again, for accuracy, there are some reporting threshold regulation that there's some concentration level that everybody comply to, right? So these are highly documented in the guidance and also in some of the white papers. Very briefly on precision, you're expected to do 3 types of precision. Repeatability, there's 2 types of repeatability: system and analysis repeatability. Okay, and then intermediate precision by different analysts, instrument, and reproducibility, which is can be used some of your method transfer data or by using interlaboratory trial, right? So there's a lot of detail of the expectation, right? I just want to say that for drug substance is a very tight specification, right? Typically, you're expected to release the specs of 98% to 102% potency for the drug substance, right? So your analysis repeatability for drug substance have to be really tight, have to be less than 1% for the overall analytical repeatability. The RSD have to be, if you do it 6 times, have to be really less than 1%. Otherwise, if your precision for the assay is 2%, then your chances of failure is pretty high without the actual material being our specification. Okay, so this is why we really need to use UV detection for quality control of drug substance. Right? Drug products are much more forgiving because the specs are a little wider, about 90% to 110%. So the analytical precision for the entire analysis, including sample prep, is typically about 2%. You don't want it to be too wide, right? Meaning the weighing typically is the controlling factor. A little bit on the method linearity. Okay, so here is one assay where we show a very good linear correlation, 0.999%. But this method actually have a bias because the percentage of y-intercept is 7%, right? So it's something is not right there. Maybe there's some absorption going on, some interaction, right? So this have to be investigated, right? So a lot of company has an SOP that stipulate, you must have a y-intercept less than 2%, which is a good scientific risk-based approach. Just show you some sensitivity case study, right? LOQ, which is a limit of quantification. A signal noise ratio of the peak is about 10. And limit of detection is signal noise ratio is about 3, right? This is the simplest way. I prefer this way. There's a 7 different way being documented by the USP to calculate the limit of detection and limit of quantification. So I always use the simplest way, which is based on signal noise ratio. There's different way to define the noise. I prefer to use the simplest, which is peak to peak, right? Your CDS, your chromatography data system, actually give you many different ways to calculate noise. Okay, finally, some case study on actual validation summary data, right? Once again, I encourage you to read the white paper, which give you a little more detail documentation on some of these things, right? So this is my phase 0 early phase method, which I developed, right, at Purdue Pharma in 2015, right? And here's the validation data, right? Now, at that time, I was not experienced, right? So I overdid some of that data, right? Meaning that using a wider range, I actually do a robustness, which is not needed at phase 0. Robustness is not really needed until later than phase 2, right? But this is a whole set of data which show you, demonstrate a validation from a method development to validation, right? So this is actually a good example, which was documented in the book on how it was being done. And I used a different DOE study to do robustness. And so it's just a good case study, even though that I actually exceeded the expectation for the regulatory guidances, which is usually not a good idea, right? Because you do spend more time doing that, right? But here's a set of data which is useful. Another set of data which is actually came from Gilead Science for that very complex sample with 60 retention marker solution. And they submitted that to the FDA. It was approved. The method was fully validated, used for release testing and stability study for that HIV standard of care combo product, right? So we published that in also the LCGC vaccine in 2014. Here is a summary on how the type of data that was obtained, looking at the validation of the API and some of the impurity, right? Okay, so here's a reference list on, so once again, some of the one in red are the one that's probably the most updated, right? So chapter 10 and 11 of the book. I'd like to point out this particularly a white paper on separation science and drug development, the quality control aspect of it. It's a really nice white paper written mostly by Darwin Call, my ex-colleague at Genentech, and myself. And Darwin did a really good job on that. This is white paper number 4 of that year. Number 3 is on analytical development. I want to point out 3 white paper that was focusing on stability study and testing of pharmaceutical, and also development of stability indicating method, which is published in August issue of LCGC magazine, and method validation. This is one of the best white paper I ever written. I spent over 100 hours putting everything together, actually even more detailed than the book chapter, right? I just checked the website. It was published today. You can download it. I'm going to put out the gallery into my website, right? So you can actually get a better printed copy available on my MWD Consulting website, right? So that was just published today, actually. So here's my final summary of this webinar. I want to mention that most stability indicator methods are based on reverse phase UV method under grading condition. And I explain some of the basic reason. Okay, as a scientist, it's nice to be able to explain in simple word why the rationale that you do something, right? It's good to have a preference, but you need to kind of have sufficient verification or justification of your choices. We very briefly review the 5-step traditional method development approaches using a methodology called selectivity fine-tuning by changing the illusion profile and resolution, fine-tuning the resolution of some of the critical pair which are partially resolved, right? So everybody do this some of these things, even though they might not call it selectivity fine-tuning, that's what they were doing. I postulated the 3-pronged template approach. So anybody can emulate some of these approaches to expedite and give you much more consistency across the board from a large organization, right? Particularly useful for early clinical phases and for new drug development. Finally, I propose a generic gradient method, a 2-minute method, universally applicable using the best column technology, optimize operating condition and the dimension, using very simple mobile phase. Even without phosphate buffer or ammonium formate buffer, one can get very good peak shape with very highly basic peaks. And appear to work really well for potency assay for many NCE. And the generic method can be adjusted very quickly for stability indicating application, right? It's really definitely serves as a good first step of the starting point of any method development for pharmaceutical sample. And then later on, once you understand how things work well with your compound, you can always go to a longer column, a different binder phase or different mobile phase. You can change all that later on. So the take-home message that a lot of time, simple is better. Okay, always pick the simple approach, time-saving approach in most of your endeavors, even including very sophisticated scientific investigation. Okay, finally, I'd like to acknowledge Ernest Salkal and also Jack Shaw from Welsh Material Incorporation for sponsoring this free webinar, allowing us to deliver this at no cost to the attendees. I want to mention that the PDF file of the slide deck would be sent to all the registration guests by Welsh within next few days. I sent a copy to Jack, and up to him when and how that was being sent to the registered guests. I also asked him to post that at welsh.com at the Welsh website for a month or so, right? We can have that over, or you can download that beginning next week my own website, which is www.mwdconsulting.com, right? I'm going to have it there for at least up to the end of the year, right? So after that, I might decide to pull it out. Okay, I'd like to ask all the attendee, feel free to connect with me at LinkedIn. I have close to 10,000 followers, and most of these are pharmaceutical scientists with a focus in HPLC of chromatography. And here's the link to my profile, right? So definitely send me an invite. I'm usually very open to connection over there because it's nice to have a community that we can share idea and also promote the best in science. I want to mention that I have negotiated with John Wiley to have a discount code available if you order this book directly from johnwiley.com, right? You can also order it from amazon.com. The pricing is better if you order directly at Wiley. So this code allows you to have 30% off. either the ebook, the electronic book, or the paperback. Right? The paperback listed price at $100. So you can get it for about $30 off, $70 plus shipping. Also, I want to mention that some of the paper that was published today you can download that. You can download a free book sampler of the book. You can look at chapter 1 for free, and also the detailed discount ordering detail from a book sampler, which is available at the website, right? And also any paper that you see, which is not copyrighted, I have put it. I put up 100 of my white papers published on the website. So feel free to download that. Okay. So on that note, I'd like to thank Ernest for being a generous co-host and for being a great moderator. I'd like to thank you for attending it in your busy schedule. I know that there's tons of competition in there. Hopefully that 2 hours worth your time, right? So I'm going to ask Ernest to help us do the question and answer period, right? Should I unmute everybody, Ernest, and let them just add? Okay, all right, so let me do that, right?
Okay, can you hear me?
I can unmute, or yeah, I can hear you, yeah, okay.
Oh, okay, good. I hope
everybody's unmuted, right? So you're welcome to use your video. You can just turn on your video so we can get a look of you. It's always good to do face-to-face, right? And also when you're talking, you should unmute yourself, right? You should be able to unmute yourself because I have unmuted all of you. So just click on your microphone and unmute yourself. You want to ask a question, right? But please feel free to say hi and network with each other, right? And we have about at least 15 minutes to allow you to ask any question, right? Okay. So Ernest, please.
Yeah. Okay, Michael. We have 3 questions. I have one of which is mine.
Okay.
Aditi, she's new to method development, and she wants you to explain how chiral chemistry plays a role in method development.
Okay, so Aditi, right? Hi.
Hi.
Yeah, thanks to allow us to see what you look like, right? Not that we need to do that, but it was always good, right?
Yeah,
okay,
I know.
So chiral are very important, right? So I spent about 10 years at Genentech, and I have 3 major projects. All 3 projects have a molecule with 3 chiral center, right? So you definitely, in your process chemistry, you have to control the stereochemistry, right? If you have 3 chiral centers, you have a stereoisomer. You have to make sure that that you make the right molecule, right? So absolutely, otherwise it would not work, right? You will not be able to go to clinical trial, right? And the analytical chemists have to deliver the quality assessment methodology that allow you every step of the way, right? So when I support the AKT inhibitor project, I developed 40 methods. Most of these are HPLC method. In the HPLC method, half of them are chiral, half of them are achiral. I use the achiral method as my primary POD method. I only use the chiral method for the stereoisomer. Because if you use both to report your impurity, you might get different answer, right? So it's very important not to pick and choose, right? So you just have to define what it is, right? Do I answer your question?
Yeah, kind of. Yeah.
Kind of, right? I mean, you can go over that. Please connect with me in LinkedIn, and you have any question, including any. I'm also a career consultant for ACS, right? So I'm very open to helping young chemists or older chemists to further their career, right? So
right. But I just want to say this was very, very helpful. I haven't done any method development, like not much. I've helped, but not done it myself. So it was very good information to start.
Right. It's very fast, right? I mean, it's also very focused to pharmaceutical development. Are you organic chemist?
Yeah. Yeah, I mean, I'm helping as a contractor with a scientist. So
okay, all right, terrific. So thank you very much for being brave enough to show your face and also to ask the question. I know analytical chemists are typically very shy, right? It's okay to be shy, but in this professional organization, it's very important to network, right? It's important to have a vast network, right? But you never know when you're going to be laid off. Okay, I myself was laid off at least twice, right? And having a strong network has become important. Now, after you get your first job, it becomes very easy, right? Because analytical chemists are very much in demand. There's a shortage for good analytical chemists with experience. So you can get really good job with high pay, high stress, GMP and everything, right? Yeah. Okay, next person, Paul, right? Yeah. Hi.
Yeah.
Hi. Thank you very much for the very interesting talk.
Right. Sure. Yeah.
May I add a question?
Sure. Yeah. Yeah. No, you're on the board. Yeah.
Yeah. Yeah. I would like to ask you about when we do methods validation.
Right.
It's very unclear for me. How can we do a robustness test?
Right. Right. Okay. So robustness are difficult. Most people use DOE. You can also buy a piece of software called the QBD, and you can do robustness study. You can do it one factor at a time, right? So I would not try to explain it here. It takes too long, but I would download the white paper. Okay, it's published today, right, from LCGC magazine, right? Take that link, and that white paper actually explain not too much on robustness, the entire process of method validation. Yeah. Okay, robustness you don't have to do until later phases, phase 2 and phase 3, right? So I mean, it's nice to know the entire process. On how do you do the whole thing, right? I know robustness, you might just worry quite a bit about it. It's not very well explained, right? But that white paper actually give you some example, right? Some of the book chapter would also give you some example on. So book chapter is really good. The book chapter from Henry's 2005 chapter actually explain different way to do robustness. But some of these are outdated, right? There are better way to do robustness. Yeah. Okay. Look at my example too. How I did a very simple example of doing robustness, column to column with precision. Robustness actually is done during method development too, right? If you develop your method development with clarity, you pick different column. You actually are doing some robustness there already. If you learn how to control your parameter using good principle. Actually, part of the robustness is the understanding of the chromatographic principle, right? So being good in HPLC, I know everybody do a lot of HPLC, but to know it very well is a different category, right? I mean, there's always different level of doing it quickly and also doing it right? Okay, yeah. All right. Well, feel free to connect with me. You have other questions, right? I can guide you. Okay?
Yeah.
Yeah. So other question? We have at least 10 minutes more, right? So thank you for Zoom for letting us go over time, right? Yeah.
Michael. Yeah. Aditi's question, and then followed up by the gentleman's question about robustness. I often found this to be a very difficult problem, particularly when transferring from lab to lab. So I got to thinking for preparation of like the mobile phase or the control of pH. I used to find that oftentimes when methods were transferred from lab to lab, that's where we got into trouble. Is there a guideline as to maybe a percent change in the mobile phase and its effect on resolution, or a suitability standard in terms of what's acceptable and what's not acceptable?
Well, actually,
is there a good place to find that information?
Yeah. I mean, there are a lot of paper. The book chapter actually referenced some of the paper on robustness, right? I think people went overboard to do robustness, right? Now, actually, there's a lot of controversy how much you can change, right? What is the method adjustment, right? the easy way to explain that is if you have a gradient method, you're really not allowed to do a lot of adjustment, right? You know, the pH could be very, very, very important, right? So you shouldn't deviate. You should be as accurate as possible, right? I have a method I developed for Purdue Pharma, which is high pH, right? The pH have to be adjusted plus or minus 0.05. And I've used turn on the temperature control, right? So a different method have different weaknesses that you have to know or where is your critical parameter, right? So it's very difficult to say what is adjustment, what is allowable, right? If it's an isocratic method, then you have a wide span, right? The USP allow you to do a lot of adjustment. But be careful when people say that, oh, you're allowed to do these adjustments without validation, right? If you talk to the FDA reviewer, right, which I'm quite familiar with some of the FDA reviewer, you definitely, you don't want to change anything unless you have done a partial validation, right? If you change anything, like going to sub 2 micron from 5 micron, you know, you have to do the validation. Otherwise, you get yourself into trouble. You don't want to be the one that get a 483 from the FDA, right? So regulators are very, you know, they are here to find, they like to pick on different things on that, right? So definitely try to follow the method, right? I wouldn't try to deviate from the method. Now, you know, sometimes from lab to lab or calibration, you can have a little bit deviation, right? So if you develop a good method and the column is very stable, most columns are pretty good batch to batch now, right? Because using high purity HPLC column, right? Most silica, and that's the most fundamental difference between 20 years ago when people don't have high purity silica. Now everybody use high purity silica. Everybody try to make their silica from scratch, right? So that really helped with batch to batch reproducibility. So I don't think we have that problem too much, right?
Okay.
Okay, yeah.
There's another question. Let's see, it's about the linearity that you showed, the correlation coefficient. Can you go back to that slide? Do you have that capability?
Yeah.
I was wondering if the R was R squared.
Okay, yeah, that's
there's an R squared there, yeah.
It is an R. Oh, it's an R. Okay, okay. So R and R squared is actually just for this case it's very, very similar. I'm not a statistician. I like to use the simplest thing, right? You know, I don't really know too much difference between R and R squared. Some people like to use R squared. Other people use I think R squared probably is the correct term to use, right? Right. Once you talk to your QA guy, those guys, they are expert in picking on little things and the little typos. If you screw up the decimal point, the signal figure, man, you know, you never hear the end of it. It's like the end of the world, right? I think sometimes some of the regulator are bureaucrats, right? I don't have you know, people doing the lab work, they don't agree with some of the QA folks. They like to pick on things that they can pick on, right? Which is, if you sign the day wrong, you can be fired, right? These type of thing, right? So I think I prefer the science-based and risk-based. You know, how much risk is there to the patient, right? If I make something wrong, it's at the end of the world, right? I know, you know, for me, it's easy for me to say. I'm not a regulator, you know. I don't support multiple projects, right?
Patricia, yeah. if you're out there, does that answer your question?
Yeah, yeah. I don't really know. You know, I like to use R, right? But I think most people use R, right? I'm not a statistician. I don't believe too much in statistician. I said that if you cannot see the difference, people use statistics to justify all kinds of things, right? Yeah.
Go ahead, Patricia.
Okay, thank you.
Where are you from, Patricia? Yeah.
I am from Colombia, but I know I'm in Canada.
Are you from Bogota? Yeah.
No, from Pereira.
Oh, Pereira, yeah, okay. Yes. A good thing about Bogota is a beautiful city.
Yes, it's beautiful.
Yeah. So does it help you? I mean, I think most people use R square, right? People that, yeah. But it depends on the SOP, right? A lot of companies, they do something a little different, right? But if you get 999 for API,
looks pretty good. 99, yeah. Your linearity looks good.
Now, Now for impurity, they're different, right? 0.98 would be sufficient. And the range are different, right? So every company had different guidance, right? That white paper actually spelled in glorious detail, right? More detail than I would like to follow. So just follow some guidance. Right? Now, everybody interpret the guidance very differently. That's the confusion. That's why when we write this white paper, I constantly argue with Kim. Kim is one of my co-authors, right? She's a consultant. She's very proud on knowing all these little detail, and we always disagree because I'm more early development person, right? And finally, we have to use a referee from our buddy from the FDA, who was there for about 20 years, chief review scientist, and she also have different opinion. She's the referee to when we get into a impasse, of which one is the right way, right? Quality people, you can argue until the cows come home. So very important for analytical chemists to understand the regulation so you can push back professionally. Because people always ask you to do more, right? Because it's free, your services are free. They don't value it. They don't care. You take 200 hours to do something. They don't care. They want it yesterday, right? They want stability done yesterday. They want you to put every batch on. So it's very important for you to understand the regulation and to push back intelligently. You cannot say that I can't do that because my cat was sick or my kid is not feeling well. You have to, you have to cite some of the IQ consortium, right? So that came from experience, right? Also, you want to be, you want to help the company to get things done too, right? Because you're on the project, and nobody knows everything, right? So a lot of time is you have to make a definitive choice, under a lot of time constraint, right? So I'm a big fan of the ex-CEO of Genentech. His name is Art Levinson. He's now the chairman of Apple after he Roche took over Genentech, right? Now, he always say that in drug development, it's very important. The ultimate guidance is what is the effect on the patient. Okay, you're not there to make somebody's job easier so they don't have questions from the FDA. Your job is to ensure that the patient's safety and efficacy is being safeguarded, right? So you have to know, so the analytical chemists actually have a very important job, which is to make sure that, you're protecting the patient's safety, right? So ultimately, we make a lot of decisions very quickly. We don't have all the data, but you have to make a intelligent decision under a lot of time constraint, right? And understanding regulation and the impact is really important, right? Okay, any other question from the floor?
Michael, we have time for, I think, for one more question.
Okay, so last question.
Fuad Amer asked the new USP General Chapter 1210, Statistical Tools on Method Validation. Are you? I don't know. I'm not familiar with it.
I'm very poor in statistics.
Comment on that?
I'm very poor in statistics, right? I definitely, somebody who want to use the simplest way of doing things, right? You look at my approach to LOQ, right? I want to use the simplest way. There's 7 ways to do that. Sometimes people kind of go into detail on things that they are good at, right? Some people are great in statistics. Other people are a strong believer in QBD, right? Now, all these things takes time, right? And also takes resources, which we might or might not have, right? So definitely, you have to do the minimum job to be in compliance, and then you have to be time, and you want to do things efficiently, right? So definitely, I'm a poor person to ask about statistics. Yeah.
Okay. That's about it for today.
On that note, I'd
like to thank everyone.
Thank you very, very much for attending in your busy schedule, and hopefully you get something useful out of that. And I try to put up my slide deck on the website, and also I think Jack should be able to send one to you, right? If you attend this one, right? Yeah. Now we have about 400 people signed up, but unfortunately, when you give it away for free, right? People don't value that. They sign up and they don't show up, right? So how many people we have actually attending? About 45, right?
There's,
okay. So right now we have right now.
Right now we're 25. Right
Well,
now.
that's yeah. So you can see that, right? Yeah. But anyway, it was good to spend 2 hours with all of you guys. And the Zoom video is a good platform, right? I really enjoyed it. Unfortunately, I'm not very good. I was stumbling around when I tried to mute everyone, right? So sorry about that snafu, right? Yeah, but we should get better and better. Yeah. Okay. On that note, thank you very much on behalf of Welsh. And also Ernest and Jack. Thank you very much, and talk to you later. Please lend me in if you want, and if you want any advice, just explain to you where we have met, right? Okay, bye now.
Okay, thank you, Michael.
Thank you, everyone.
Bye-bye.
Thank you.
Right.
Bye, everyone.
Thank
Bye-bye.
you.
Okay, any chat, we can stay online, right? And then
Yes.
Oh,
we
okay.
can,
If
we
you
can
like,
chat a
yeah.
little bit more, right? Okay.
Okay.
Why not we use that platform, right? Yeah.